And dear Loris, dear President of the Royal Academy of Sciences, distinguished guests, ladies and gentlemen. My name is Changpan Hammarström. I'm professor in clinical immunology at the Karolinska Institute and also a member of the Krefeld Prize Committee. It's my great pleasure to introduce today's first speaker for the symposium, Professor Christopher Goodenough. Together with Professor David Namazi, he was awarded this year's Krefeld Prize in polar arthritis for the discovery of functional mechanism for B-cell tolerance. Professor Goodenough was born in Hong Kong, grew up in Washington, D.C., US, before moving to Sydney, Australia, as a teenager. He studied veterinary medicine and surgery, as well as immunology in University of Sydney, and also DNA technology in Stanford University. After a PhD degree awarded by University of Sydney, he moved to US and worked as a faculty in Stanford University Medical School and Harvard Hughes Medical Institute. In 1997, he moved back to Australia and worked as a professor and the founding director of the Medical Genome Center at Australia National University. And in 2015, he joined the Gavin Institute for Medical Research, became as a director in 2018, and currently, he holds the Bill and Patricia Richter Foundation Chair at the Gavin Institute and is also professor at the Cellular Genomic Futures Institute, University of New South Wales, Sydney. When Professor Goodenough is a young student, he became interested in self-tolerance, particularly regarding B cells. It has been proposed that the self-tolerant B cells can either go clonal deletion or being rendered tolerant. But due to the rarity of this specific type of self-reacting B cells, it had long been a challenge studying the fate of these self-reacting B cells. Inspired by the cloning of T-cell receptor and the TCR transgenic mouse work in Professor Mark Davis' lab at Stanford University, Professor Goodenough had a bright idea to generate a double transgenic mice, as illustrated here, by mating two types of transgenic mice, and the first one expressing a normally foreign protein, hence egg lysosome, and in a way that can present it as recognized as a self-antigen. And the second strain mice carry a rearranged heavy and light chain genes and leading to production of anti-lysosome antibodies. So by studying this double transgenic mouse, Professor Goodenough is able to show that actually majority of B cells, the self-reacting B cells, did not undergo clonal deletion, but rather became functionally silent and producing no anti-lysosome antibodies. So this transgenic mouse work has laid the foundation for our understanding for B-cell tolerance, a very successful PhD thesis for Dr. Goodenough, and numerous influential papers from his group, and as well as important insights for B-cell tolerance. And more importantly, this has also laid a foundation for a conceptual framework for our understanding how failure of B-cell checkpoints may leading to the development of autoimmune diseases. So at Monday, Krefeld Days, some of us are there. So Professor Goodenough gave an amazing lecture in the morning for the scientists. And the afternoon, we also met the patients who suffer from various types of autoimmune disorders. And he tried to explain his work in a popular scientific way. And also he shared personal stories. So I think many of us in the audience there were touched and get inspired and just appreciating that how important basic research is. It took very long time, but eventually it may help our patients and the family. I would just like to mention one additional important papers, and where it published in 1997 in Nature, and the first author was Suzanne Hartley. I hope you're in the audience. Yeah. So I mentioned this paper because it shows elegantly that both Chris and David were not wrong, or in other words, they were both correct. So when membrane-bound self-antigens will induce clonal deletion, but soluble self-antigens will lead to energy. So this is nicely bring a piece for the two laureates here today. Finally, I just want to mention that in addition being a passionate scientist, Dr. Goodenow is also a professional surfer. And his passion and love for surfing has led to discovery for other areas, may also probably get a prize. And the citation could read as for the discovery of one of the best surf breaks in the world, named Makaronis, Indonesia. Without further ado, I would like to invite Professor Christopher Goodenow come to the stage and deliver today's first lecture for this symposia, entitled B-cell tolerance, when it fails and when it kept.
Thank you so much. Thank you, Pan. That is a lovely introduction. Thank you all for coming. This is, of course, a great honour. On behalf of David and I, tusen tack. I can't say enough thank yous to the Crawford Foundation, to the Royal Swedish Academy of Sciences for this enormous honour. Thank you for celebrating what I think is one of life's most beautiful and essential processes, B-cell tolerance. And I think we're in for a wonderful day today. We're going to hear from a series of speakers, some of whom I've not heard before, and I think it will blow our socks off. So let me start by showing you a map of the Pacific Ocean, and to mention that I've had the privilege and the luck to be part of the B-cell tolerance story thanks to three special mentors. Now, the first one was Gus Nossel, the original single-cell B-cell pioneer and tolerance pioneer, who I started my PhD with at the Walter and Eliza Hall Institute in Melbourne. The second is Mark Davis, whom I wrote to before I'd started my PhD, and he kindly agreed to my proposal to work in his lab free for 6 weeks to learn molecular biology before I started my PhD. I ended up staying for the better part of a year, and Mark eventually started paying me. Most importantly, Tony Baston. Once I'd designed this crazy PhD project that was much too ambitious, the critical thing was that it needed two things. It needed not just really great cellular and clinical immunology environment, which Tony had created in Sydney, but the ability to make transgenic mice, which was a very new technology, as I'll talk more about. And there was only one place in Australia that knew how to do it, and they were in Sydney. And so Tony was very kind to bring me under his wing and allow me to fly. You know, I wouldn't be here without him, and Tony's here in the audience. Thank you, Tony. Okay, well, before I start on B-cell tolerance, I do need to tell you about something you may not have heard of, Planck's principle. Not Planck's constant, having to do with the wavelength of light and energy, but the principle that science moves forward one funeral at a time. And you can read there why he said that. That will resonate, I think, with each of us to some extent. But in my lived experience has been different from that. I think intolerance and autoimmune disease science has moved forward one birth at a time, birth of new technologies. And I think that is one of the reasons why this prize is very special to me, and I think to the field, is that, of course, as you just heard, Holger Crawford's entrepreneurial success was entirely built on new technologies. Tetra Pak, new technologies for packaging fluids, and most importantly, new technologies for scalable dialysis machines around the world that saved hundreds of thousands of lives. So I think, celebrating how technology makes the world better and makes better health is you couldn't have a more special occasion. So thank you. What's B-cell tolerance? Well, very simply, it is how our body avoids making antibodies against ourselves. And it really is a process of dazzling beauty, but it's a process that's taken a very long time for that beauty to be revealed. In fact, over a century. Really, you can see this beautiful quote, very famous, from Paul Ehrlich in the very early days of trying to understand antibodies, where he recognized that there were certain contrivances by which the body avoided making autotoxins. We would now call them autoantibodies, because those would constitute a danger threatening our health far more greatly than any exogenous injury. Well, what is that process? So that's the process that's taken so long to come to grips with. But I'll take you very briefly through some of the key foundational steps that tell you both about how the field moved, again revolving around technology, but also telling you why it is important. So the B-cell tolerance first came into view back with the subject of the first Nobel Prize, Emil von Behring's huge advance of serum therapy for what then was a terrible disease, diphtheria, which, as you know, involved giving plasma containing IgG from horses that had been hyperimmunized to diphtheria toxin to children, ideally in the first 2 days of a diagnosis of diphtheria. Now, that was a dangerous treatment, even though it did save hundreds of thousands of lives at the time. And what made it dangerous is serum sickness. So serum sickness, the problem is that when you inject horse immunoglobulin into a human, our immune system, our B cells and T cells recognize that as foreign, and the B cells only make antibodies against the parts of the horse immunoglobulin that are different from our own. And those antibodies, in about 8% of people that are given horse serum, form complexes in the bloodstream, which lead to vasculitis and often quite severe damage either of the skin, the nerves, kidneys, heart, etc. Now, if you give human immunoglobulin to people, that doesn't happen. And of course, in my state alone, in Australia, New South Wales, that is an incredibly widely used therapy and prevention. So we literally give patients tons of human immunoglobulin, tens of thousands of patients for hundreds of different diseases. And the way our body accepts human immunoglobulin without triggering serum sickness and without making antibodies against it, it sees it as self. It's big business. It's the basis of Australia's largest pharmaceutical company, CSL, which is now a global business, selling human IgG from plasma for serum therapy. It's not a perfect process, this B-cell tolerance. So some of us, particularly older in life, will start making antibodies to our own IgG, what are called rheumatoid factors, because they were first discovered in people with rheumatoid arthritis back in the 1950s. Now, in many people, oops, sorry, I keep forgetting to advance the slides. No, I've gone the wrong way. What have I done here? No, I've gone the wrong way. Sorry about that, lost. Now, in some of us, those rheumatoid factors will get up to a titer and a quality that they'll actually cause many of the same symptoms as serum sickness. And I'll come back to one of those diseases, cryoglobulonemic vasculitis, later. Now, back at the turn of the 20th century, the other clear evidence of B-cell tolerance was, again, a subject of another Nobel Prize, Landsteiner's discovery of the blood groups and the antibodies that we make against them. So I'm sure each of you probably know your blood type, whether you're A or B or O or AB. And the key thing that Landsteiner discovered was that we make antibodies against other people's blood types, but not against our own. And that, of course, became the foundations of safe blood transfusions, which are a cornerstone of medicine ever since. Again, that tolerance to blood types is imperfect. In some of us, we will start making antibodies against our own blood type. And that first came into view, particularly with this patient that's described here in a 1930s paper, who had what is called cold agglutinin disease. And you can see there, this person lived in a place that's even colder than Sweden, Minnesota. And in winter, his bloods would go, his urine would turn red because his red cells were lysing. And his fingers and toes and tip of his nose in cold weather would go purple because the blood vessels were blocked, because he was making antibodies against a blood type that was yet to be discovered called I for individuality. And that was clogging his vessels and lysing the red cells. When he ate ice cream, those antibodies would agglutinate the red cells in his tongue, and the tip of his tongue would go purple. Now, the basis of that, I'll come back to, has a lot to do with Frida Stevenson, who's here and we'll hear more about later. Now, so how does B-cell tolerance work? A little bit more important steps in the history. Well, an important clue came as to how you don't make antibodies against your own blood type from a geneticist in the US, Ray Owen, in 1945, who discovered that dissimilar cattle twins exchanged blood between one another through the placenta before birth and were lifelong chimeras, and as a result would not make antibodies against their siblings' blood type, even if they were totally dissimilar. And that observation led two virologists in Australia, Macfarlane Burnett and Frank Finner, to realise that this wasn't some law of nature, it was a learned process, that the body learned somehow to recognise our own blood types and our own antigens as self and not make antibodies to them. And that was experimentally demonstrated beautifully by a British team led by Peter Medawar, in 1953, where they coined the term actively acquired tolerance. And then in 1957, Burnett articulated what was an incredibly bold and beautiful hypothesis, a theory for how this process was learnt. And that process, let me just take you through it. It's beautiful both for its simplicity and daring in terms of how it leapt forward relative to the tools and technology that were available at the time. It was also very binary. So one of the brilliant ideas he had, which we know is correct now, was that blood stem cells would activate a mutator process, we now call it VDJ recombination, that would generate, mutate the antibody genes so that each different B cell made a different antibody, denoted by the different colours. And then those antibodies would be displayed as receptors on the surface of the B cells and be tested first for whether they were potentially harmful by binding to self. And in Burnett's binary vision, that was either a yes or no decision. They either bound, in which case the cells were deleted and purged from the repertoire, or they didn't bind, in which case the cells were deemed as potentially being useful and are kept and allowed to mature so that later, if you were immunised or infected, any B cells whose surface immunoglobulin bound to the virus, for example, would then multiply and turn into plasma cells and secrete antibody against the virus that would not be against your own blood types or your own IgG. Now, it isn't as binary as that, but in simple terms, you can think of Burnett's theory as following that motto of if in doubt, throw it out. And as I'll come to, that doesn't turn out to be the way B-cell tolerance works, not in that simple binary way. Well, what did happen next? So at the time that David Namazi and I came into this field, this tolerance field was actually in disarray. Gus Nossel, who I'd signed up to be a PhD student with, had just published this a momentous review of the field, which basically said the field's a mess, different camps believe different things, we don't know how it works. And the problem was that Burnett's clonal deletion theory had been discarded by most people as disproved. And Gus Nossel's modification of it had also been disproved, particularly, for example, by experiments by Erner and Joran Moller here in Stockholm at the Karolinska, who showed that they could find self-reactive B cells that would respond to mitogen quite efficiently. So that, on the face value, seemed to rule it out. And many other groups had done similar things. IJ positive suppressor T cells were very popular at the time. It occupied volumes and volumes of the Journal of Experimental Medicine. They went away the minute that Michael Steinmetz and Lee Hood sequenced the MHC and discovered there was no gene for IJ. The idiotype network was fabulous as an idea, but impossible to test and never really proved to be reproducible. And many people, as Gus pointed out in 1993, felt that tolerance was kind of dead and that it was just some kind of regulation, which really left us with no explanation for all these fundamental observations of Ehrlich and Landsteiner, Medawar and Owen. The thing that really was the problem for the field was two things: complexity and a lack of technology. So the complexity is that Burnett's mutator mechanism generates millions of different antibodies, and each B cell has a different antibody. So to see what happens to a B cell that sees your own blood type is like looking for a needle in a haystack. And if your hypothesis is the needle has been removed from the haystack, you can see it's an impossible task to actually do the test. And so what David and I were very lucky about was by the time we came into the field, huge strides had been made in technology to clone antibody genes, to sequence them, to engineer them and rearrange them, and it was starting to be possible to put rearranged antibody genes back into the inherited germline of mice as transgenic mice. And so what we did was to focus on a very model protein, Hennig lysozyme, because it was the best studied protein antigen known at the time. And we made transgenic mice where the B cells would all have the same receptor so that it would be easy to see what happened to them if they were self-reactive. Now, to make them self-reactive, we introduced the lysozyme gene from chickens into the mouse germline so that lysozyme was present in the serum alongside self-IgG as if it was another self-protein. And then we crossed the two mice together, and the effect on the B cells was as clear as day. You didn't need, well, you did need some sophisticated technology, but the effects were not ambiguous or subtle at all. They basically knocked you over. Now, that all sounds kind of simple, but it really takes a team for technology to be put together. And I was very lucky to be part of an incredible team. So let me just briefly give you a sense of that. So as I mentioned, Tony had put together an extraordinary centre, leveraging all of his deep knowledge of clinical immunology and cellular immunology at the University of Sydney. I was very lucky to collaborate with Sandy Smith-Gill and Tom Lavoie at the NIH, who were really the world's experts on monoclonal antibodies to Hennig lysozyme and had made a series of monoclonals and cloned the genes that they provided as a collaboration. Now, to make transgenic mice, there were really only a few centres in the world that could do that. The best was, one of the best was in New York, Frank Costantini and Liz Lacey. And just lucky for me, Kathy Raphael had just finished her postdoc with Costantini and Lacey and had come back to Sydney in order to set up an operation making transgenic sheep. And she kindly agreed for me to come out to her lab and learn how to make transgenic mice in the western part of Sydney. And that, without that, nothing would have been possible. Now, I was a pretty impatient microinjector of DNA into mouse eggs, and I'd killed a lot more than I tried. But Tony paired me up with a brilliant senior technical person, Jeff Crosby, who was incredibly good at injecting mouse eggs. And, I only needed to show Jeff once, and then he went off and really worked his magic. Then, once you've got transgenic mice, you still need some of the old classic methods of cellular immunology, Yerner's plaque assay, adoptive transfers, bone marrow chimeras, and Helen Briscoe in Tony's team was a master of these things. And I could go on. But the point being, it really does take bringing together many, many different old and new technologies for things to move forward. Now, what did we see? Yes, most importantly, flow cytometry. Tony had gotten a big grant from the government to get the state-of-the-art machine, which I think did 3-colour. I'm not sure. It might have only been 2-colour. 2. We thought it was the ant's pants. 10-inch floppy disks. But without that, you couldn't see what was happening to the cells. So these were the kinds of things that were critical. And what did we see? Well, we saw 2 things. When we measured the serum of mice, we saw what you can see on the right, which is when the B cells were seeing lysozyme in the serum as a self-protein, they stopped secreting the antibody against it. So that was exciting in the first step. So tolerance was happening somehow. But the really exciting result was when we put the cells through the flow cytometer, and they weren't deleted, but they'd undergone this peculiar change where one of the 2 flavors of surface immunoglobulin, IgM, had almost completely decreased from the surface of the cell, but the other flavor, IgD, was still high. We had no idea what that meant. And you immediately would wonder, and many did, that this was just some transgenic artifact, except for that most B cells in normal mice and in people have that exact same phenotype of low IgM, high IgD. And so we hypothesized at the time, and with other experiments, that actually maybe our bloodstream and the mouse's B cell repertoire was full of these self-reactive B cells that were somehow functionally sized. I presented this at a big meeting in Cold Spring Harbor in New York. I didn't even have my PhD yet, and the first question was from the great Av Mitchison, the founder of the European Journal of Immunology, one of the discoverers of T-cell, B-cell collaboration. And Av said, I don't know why you've done these experiments, because everyone knows there is no such thing as B-cell tolerance. And luckily, I read Av's papers, and I knew why he'd misinterpreted his results, and I tried to explain that to him. But it took another 30 years of experiments to try and prove that and answer Av's question. And I was very lucky that David was almost immediate, had already come out with his results, which showed B-cell tolerance. Unfortunately, David had a different result from us, which only was fuel to the fire that maybe all these transgenic mice were just leading everyone down the garden path and had nothing to do with physiology. So what did we do? Well, in the ensuing several decades, we and many others found similar results, and a lot of new technologies came along that allowed us to start to decipher the cellular events. Pan mentioned that Suzanne Hartley, who by that stage had become my wife, at least put David and I out of our misery by showing that the 2 different results were actually 2 sides of one coin, and there was a logic to that side of things. But there was a lot of work to be done. Now, most important at the core of this is that it wasn't binary the way that Burnett had envisaged it. Binding self or not binding self is not an all or none question. It is a continuum. Affinity, avidity, all of these biophysical parameters are tremendously important. And really what was happening, of course, was that cells, and we were able to show this and others to show, that cells with modest to strong levels of self-reactivity would not be deleted, but would store a negative signal. Cells that were very strongly self-reactive and their receptors cross-linked a lot would be either deleted or edited, and that's David's story, and I will leave him to tell you that in after lunch. But many of these potentially harmful cells would be kept because they might still be useful, but they wouldn't secrete their autoantibody. Now, over the couple of next decades, a lot of other new technologies were born that allowed us to piece together what this stored negative signal was. So, for example, Sarah Bell, when she was a postdoc with me, did some beautiful biosynthetic biochemistry on the cells to show that there was more IgD being made in the tolerant B cells, and it would outcompete the IgM for the CD79 subunits that are needed to come to the surface. Jason Sister, when he was in lab, used some beautiful mouse molecular genetics to show that a tyrosine phosphatase was at the core of dampening the signal and the way that the B cells activated calcium and responded to a self-antigen so that they could be kept in the circulation and not deleted, even though they were self-reactive. Then Jim Healy in the lab, along with Mike Cook, showed that there were qualitative and quantitative changes in the amount and nature of intracellular calcium and the downstream signals to uncouple the antigen receptor from NF-kappa B, which is crucial for plasma cell formation and proliferation, but to keep signaling through NF-A-T transcription factors into the nucleus, as long as the cells kept binding antigen. And Richard Glynn and later Zara Saburi, when they were in the lab using microarray technologies or RNA sequencing, were able to show that that different quality of signal that was in the tolerant B cells was activating a very different transcriptional program in the nucleus, involving transcription factors EGR2 and EGR3, turning on another transcription factor, ZFP318, which we discovered, Ansel Mendes discovered in an ENU mutagenesis screen, is crucial for upregulating, changing the splicing of the heavy chain mRNA to make more IgD, which explains why part of why IgM goes down and IgD usually goes up. And then that attenuates the response to antigen at the surface. And Etienne Maslé-Farquhar a few years ago finally connected these different branches of this puzzle by showing that that EGR2 and EGR3 are at the core of inducing ZFP318 and downregulating IgM by increasing IgD. And they also turn on a suite of transcription factors. They also inhibit a suite of transcription factors that are critical for the formation of plasma cells and a cell that's poised to become plasma cells, what's called an atypical memory B cell or an age-associated B cell. Well, the other big problem that I've faced ever since that first Cold Spring Harbor meeting is people would say, Chris, it doesn't make any sense. Why would nature want to fill the B cell repertoire with useless B cells that can't secrete antibody, anergic B cells? And in Gus Nossal's clonal analogy, the B cells, as he defined in his in vitro experiments, could not proliferate to either antigen or mitogen like LPS. But we found immediately that these tolerant B cells in the double transgenic mice proliferated fine to LPS. They just didn't differentiate into plasma cells and secrete the antibody. And Mike Cook, when he was in lab, made the perplexing discovery that these tolerant B cells were actually primed to proliferate even better than to anti-CD40 and IL-4, mimicking what the signals that a helper T cell or a follicular helper T cell would give. So that really made no sense as to why you would do tolerance this quite dangerous way. And we showed that these processes could reverse and the cells could regain the capacity to form plasma cells. But we did have this sentence in 1991, a little bit of a throwaway sentence that, well, maybe the cells could mutate if they saw a foreign antigen, their antigen receptors, and lose their self-reactivity and acquire a more appropriate specificity. And so maybe that was a rationale for why tolerance was working this way. We expanded on that in a 1993 paper in a book when we discovered that a lot of these tolerant B cells were in the follicles, right where germinal centers form and hypermutation happens. But I have to say, I didn't really take it very seriously, and we didn't have the tools at the time to test it well. And so this is an instance where Planck's principle was operating, but the shoe was on the other foot now. It was me holding up progress and not taking it seriously. Now, gradually, second and third generation transgenic mice have allowed us to come back to that in 2014. And I wanted to tell you a story which we haven't yet published, but I think connects this all together and addresses one other really important question that we always faced, which Jim Allison once said to me on the back of a bus. Jim, anti-CTLA4 checkpoint inhibitors, said, "Chris, I'm going to start a new journal just for you and your mates, and it's going to where you can publish all your transgenic artifacts. It's going to be called Shit Happens." Excuse my language, but imagine that with a good Texan drawl. This was a worry. How would you prove that any of this actually happened in people? So the way that we've come about this is to come back to that one of those very first autoantibodies, cold agglutinins, which I mentioned before, which see these chains of N-acetylactosamine that are actually on glycolipids and proteins. And I need to tell you several things about them. The first is that back in the 1960s, Henry Kunkel and Don Capra discovered that these cold agglutinins, when they're made in people, usually have the same heavy chain idiotype. And Frida Stevenson, who's here and we'll hear from, together with Virginia Pasquale, who was working with Don Capra, showed that the reason for that is that these cold agglutinins almost always use the same heavy chain V element, VH434. Frida made a beautiful monoclonal antibody back in the early 1980s against this VH434 idiotype and showed that 6% of lymph nodes in otherwise healthy people, the B cells displayed this potential cold agglutinin idiotype, but only 0.2% of serum IgM, but using very sensitive and accurate radioimmunoassays. And in subsequent papers, Frida pointed out this might mean that these cells are being regulated in the same way as these lysozyme transgenic B cells. So how are we going to test that? Well, we came to, let me just make sure I don't lose my place here. So an experiment that we have done using third generation transgenic mice really starts with the greatest vaccine of all time, the vaccine that eradicated smallpox, vaccinia virus inoculation. And so a Danish group a number of years ago took a group of people who had received the smallpox vaccine 30 years earlier, boosted them with vaccinia virus, and 14 days later used what was new technology to isolate and sequence the immunoglobulin genes from individual plasma cells circulating in the blood of these people, and then clone and express a whole series of several hundred antibodies against vaccinia. And a number of them, and that's what we got interested in, used VH434. And one that we focused in is this antibody 589 as a representative. And what we showed a number of years ago is that the 589 antibody only binds vaccinia. It doesn't bind the I blood type. But if you revert the 27 mutations that it's acquired at some point in that donor over the 30 years of vaccination to the ancestral pre-589 antibody, you now have an antibody that binds both the virus and the I blood type. And so it's representative of that 6% of the human circulating repertoire. So this is about as close as you can get to a transgenic mouse, but we still need to do an experiment in mice to take it further. And this is what we haven't yet published, but as a team effort. Rob Brink, in particular at the Garvan, using CRISPR-Cas9 technology, made a pre-589 immunoglobulin transgenic mouse by knocking in the heavy and light chains for this antibody into their correct location in the germline of the mice. And what you see if you use Frida's anti-idiotype antibody to stain is that a high proportion, about half of the B cells in these mice, express the pre-589 human antibody, and the other half express mouse immunoglobulin genes. And what you can see in red in the middle panel there is that the pre-589 B cells are all bunched up in the IgM low range of the normal distribution that normally exists. Now, that could be just because the transgene's not expressing properly or something like that. But what we also did with Rob is to knock out one of the main enzymes that creates those N-acetyl polylactosamine chains that constitute the I antigen, B3GNT2. And when you knock out the enzyme that makes the autoantigen, now the B cells are IgM high. So this is essentially the reverse of what we did with the Hennig lysozyme system, but it's the same result. They now become IgM high. Now, if you look at the serum, so if you're just looking at those red cells, if you look at the serum, what you see is that in the pre-589 transgenic mice, even though they've got enormous frequencies of B cells that could potentially make this I antigen binding antibody, there is no serum IgM from those cells. So it's essentially recapitulating Frieda's original result in humans. But if you get rid of the enzyme that makes the I antigen, now they secrete huge amounts. So we're really now connecting the two worlds of human and mouse immunology. Now, we've done single-cell sequencing of the follicular B cells from these mice and compared the ones that are expressing the human antibody to the ones that are expressing the mouse antibodies. And most of the genes that are up in the VH434 positive B cells are the same genes that we defined as being up in the HEL-tolerant B cells or down in follicular B cells where we've removed that tolerance response transcription factors, EGR2 and EGR3. So these B cells really are expressing a human autoantibody that's one of the first order antibodies. They are storing the same or very similar negative signal to the one that we've defined in this very artificial lysozyme model. Well, they're not anergic. And so what you find is that if we now repeat in the mice the experiment that was originally done that led to this plasma cell in the person, 30 years of vaccination, but now the way we do it is a bit different. We dilute a small number of these human immunoglobulin expressing B cells from the pre-589 transgenic mice, transfer them into an entirely normal mouse with a full diverse repertoire of B cells, and then inoculate that mouse with vaccinia virus. And then between 10 and 24 days later, we do flow cytometry and find that these pre-589 B cells are very efficiently activated to become germinal center B cells and accumulate there. And when we do single-cell sequencing of hundreds and hundreds of those cells, we see that they're acquiring recurring mutations, 4 of which are the same mutations that were acquired in the original donor. And so if I go back one, so by day 10, 98% of the progeny of these B cells in the germinal center have acquired at least one of these 5 recurring mutations, which, as you can see here, each of these different 5 mutations decrease by at least 50% the binding of this antibody to the self-blood I type. But they really have negligible effect on binding to the virus. But by day 16, most of the cells have acquired 2 of this set of 5 mutations and have pretty much lost their self-reactivity and have started to increase their virus binding. And by day 24, almost all the cells have trios or quartets of the same mutations that were originally acquired in the original human donor. So this is why I say this is an incredibly beautiful system. This is an evolution machine that nature has made to evolve antibodies from bad antibodies to exquisitely specific antibodies that are the kinds of tools that we use in so many different medicines and diagnostic tests today. Now, how that does that is still a mystery, and so there's a lot of work to be done. Now, there's another really important thing about cold agglutinins, and this is something that Frieda was always reminding me, and I always neglected to actually read the literature. Let me skip through this. Well, just to summarize what's happening here is that the pre-589 cells are storing a negative signal. They're not deleted. They're allowed to persist without secreting the autoantibody. And then if vaccinia virus comes along, the cells are stimulated to proliferate, hypermutate their immunoglobulin V regions, and very quickly find daughter cells that only bind the virus, and those are the ones that go on to dominate the response and secrete the antibody that's virus specific. Now, there's one situation where that doesn't happen, and that's in Mycoplasma pneumonia. And this actually, so Mycoplasma, you know, is a cell wall-free prokaryotic bacterium that normally lives in our airways and causes pneumonia. And back in the 1940s, there was a beautiful series of papers, mainly from soldiers, when there were big outbreaks of what was then called atypical pneumonia. The organism hadn't yet been identified, where it was different from any other viral or bacterial pneumonia because these individuals 69% of them would develop cold agglutinins in their serum, which you can see there. And some of them, about 8%, the titers of those cold agglutinins were sufficient to cause autoimmune hemolytic anemia. Now, what's happening? Why is this one organism breaking tolerance so well? And the reason seems to be explained, I think, by Tenfasi back in the 1980s, who showed that the way that this organism adheres to our own cells and to our airway is by having receptors that bind to silated forms of the I glycan, the N-acetyl polyacetyllactosamine. And what Tenfasi showed was that the organism actually gets coated, acquires those self-glycans. And so now what the B cells are seeing is essentially incredibly immunogenic foreign particles with toll-like receptor ligands, immunogenic T cell peptides from the microbe, but the B cells, 6% of your B cells are going to see this microbe through a VH434 receptor that's a cold agglutinin. And there's no way that when they go into the germinal center, they're going to find a mutation trajectory to mutate away from binding self and only bind the microbe, because they are binding self on the microbe. Now, there's another situation where that problem occurs, and this brings us back to that other autoantibody that I was talking about before, rheumatoid factors, and that's in people with hepatitis C virus. So as you know, people with hepatitis C virus are chronically viremic with millions of virus particles per mil of blood at any one time. And it's well known that those particles are decorated with our own IgG, with non-neutralizing IgG. It's one of the clever things about this virus. So that what happens is 50% of people with chronic hepatitis C virus develop a rheumatoid factor that has sufficient affinity and problems with solubility that at 4 degrees it crashes out of solution. And so it's what a laboratory test would call a cryoglobulin. Now, usually that doesn't lead to clinical symptoms, but in 10% of people, the titers and avidity and solubility of that antibody is such that those immune complexes actually crash out of solution either in cold extremities and cause a purpura, or crash out of solution in the mesangium in the kidney, where water is being rapidly sucked out across Bowman's membrane to cause glomerulonephritis, or to crash out in the nerves and cause neuropathy. So what we've been doing, and have just recently published, is to try and piece together how the virus is breaking tolerance so efficiently for this one particular virus and triggering pathogenic rheumatoid factors. And without going into the details, which are published, Clara Young and a very large interdisciplinary team started working, again, very similar to the VH434 story, but again, another of Kunkel's discoveries from the early 60s was that 60% of these pathogenic rheumatoid factors that are cryoglobulins use the same heavy chain V region, same idiotype. There's a different one, VH169, and in this case, this Y idiotype, as Kunkel called it, does require a particular light chain, a kappa V320 light chain as well, and as I'll show you, a particular length of CDR3 in the heavy chain. Now, what's interesting about hepatitis C virus is 90% of the neutralizing antibodies you make against hep C also use this VH169. So the temptation is to say, well, this autoimmune disease is antigenic mimicry, that the virus E2 protein is triggering antibodies that then cross-react with IgG. And so what we did by looking at the unmutated ancestors of a whole series of hepatitis C virus E2 antibodies or IgG antibodies that use the same VKappa 320, so they're paired for CDRs 1 and 2 on both heavy and light chain, was to see that they're different. The rheumatoid factors have short CDR3s in the heavy chain. The hepatitis C virus ones have long. And of course, that's set during VDJ recombination in a pre-B cell, as David will tell you more about. And what we were able to show using some methods to detect even very low affinity binding is that the unmutated ancestors of the rheumatoid factors only bound IgG and had no detectable binding to E2, whereas the unmutated ancestors of the virus antiviral antibodies had no detectable binding to IgG but only bind the virus. And that's presumably, structurally, that makes sense from what's known of the crystal structures of these two kinds of antibodies, that the different CDR3 length is pivotal for the differentiating between those two antigens. Now, what we did, we couldn't measure for the unmutated ancestors binding to IgG by these unmutated ancestors of the rheumatoid factors, and we went to a method that had been developed at the Vaccine Research Centre in Bethesda, of expressing membrane IgM on the surface of transiently transfected HEC293 cells, and then multivalent soluble fluorescent antigen. And when you do that, what Clara discovered was that you get cells with a whole range of IgM levels, and there's this striking effect where only above a certain density of surface IgM do you suddenly see binding to IgG. And in collaboration with Dan Frenkel, and Stefano Angelo Uberti in the UK, and Dan really solved this biophysical question and explained these step functions like this. It's a phenomenon called superselectivity, which is mathematically understood. It's not simply avidity. But using that, we've actually been able to estimate the affinity of these ancestor rheumatoid factors, and you can see they're down in almost millimolar affinities, incredibly low affinity, and that presumably is one of the ways that these cells evade these tolerance processes, is that they start out in the bone marrow with such low affinity and such rapid dissociation that the IgG in solution, which is a monomer, probably stays docked to the receptor for a half of a second at best. But when a virus is decorated with multiple IgG Fcs, that as long as there's enough surface IgM, this biophysical principle of superselective binding crosses a threshold and engages a bunch of receptors on the B cell and transmits a signal to activate it. And so what we have done is, for 4 of these patients, is a whole genome sequenced the B cells that are making these pathogenic rheumatoid factors. And what you find is that they have acquired many mutations in the V regions of the antibodies, but in the course of that, they've also acquired thousands of mutations across the rest of the genome. And in fact, the same burden of mutations as you'd find in a chronic lymphocytic leukemia, less than you'd find in a non-Hodgkin's lymphoma. And but that's similar to the burden of mutations that are acquired in normal memory B cells in someone over the age of 50, which each of these patients are, which Peter Campbell's team has shown that come from 2 types of mutation that accumulate in all of our B cells. And that's the same we see in these pathogenic B cells. Half of the mutations have this mutation signal called SBS blood, which appears to be a clock for how old your hematopoietic stem cells are. And then almost another half are this SBS9 signature, which is thought to be off-target mutagenesis by the enzyme that does the somatic hypermutation of the V regions, AID. Now, most of those mutations are irrelevant, and in normal memory B cells, they fall in innocuous places. But in each of these clones, at least one of those mutations creates a classic gene that you'd find recurrently in lymphomas or CLL, either often activating the NF-kappa B pathway. And we can often, by creating clonal trees using the V region mutations, see that these lymphoma driver mutations, which are not sufficient to create a lymphoma or leukemia, are acquired relatively early in the ancestral tree of these pathogenic clones, or what we call rogue clones. Now, so what we think is going on here is, putting that all together, is the explanation for autoimmunity. Hepatitis C virus illustrates what a dangerous game the system is playing. So VDJ recombination, which is a form of somatic mutation, in 5% of B cells creates a VHVL combination CDR3 link that binds weakly to our own IgG. Now, if you have millions of particles of virus coded with IgG, those B cells get stimulated to proliferate, to start to mutate their antibody and improve their affinity somewhat, creating potentially a cryoprecipitating antibody. And they also acquire thousands of mutations across the genome. They make, because so many cells are being activated, you get 50% of people make a rheumatoid factor cryoglobulin, but it's usually not in pathogenic quantities. But then one of the cells acquires a driver mutation that gives it a growth or survival advantage, and that's enough for it to make more progeny, more plasma cells, more immunoglobulin, and you cross a threshold where you now have purpura, vasculitis, glomerulonephritis, neuropathy. Now, we've done this kind of thing now for quite a few patients, both with hepatitis C virus, with Sjogren's syndrome, and also in unpublished studies in myelin-associated glycoprotein neuropathy. And in each case, finding these rogue clones making the pathogenic autoantibody in each case with one or sometimes two classic driver mutations, often activating the NF-kappa B pathway. Remember, one of the ways that the cells stored a negative signal was to uncouple the NF-kappa B pathway from the antigen receptor. And so this is, again, an experiment of nature that tells us that what we were seeing in the mice is relevant for people. Now, in my last slide or two, I just need to link into the talks that I think we're going to hear this afternoon. What we noticed, once we could track these clonal trees that were making pathogenic autoantibodies, we noticed quite a number of these patients have been treated with rituximab, with anti-CD20 B-cell depleting antibodies, ineffectively. And for example, this patient, where the activating mutation in the rogue clone is a classic CARD11 gain of function, activating the NF-kappa B pathway from the androgen receptor, over half the cells in the clone by flow cytometry don't have CD20, so they can't be touched by the depleting antibody, but they express CD38, and by gene expression programming, using the new tools of single-cell RNA sequencing, these cells are all plasma cells, mostly out of cycle. They're not plasma blasts, but they can't be depleted, but they still have surface immunoglobulin, they still have MHC2. And so the explanation for why rituximab doesn't work in these patients is not that it's not a sufficient depth of depletion, it just doesn't hit all the bad B cells that are causing the trouble. And in fact, when you go and look into the literature, of course, the field, we've always liked to think, many of us, Av Mitchison, one of the reasons he didn't like B-cell tolerance, he was very T-cell centric. And there's this idea that we have a command and control structure in immune regulation. The dendritic cell activates the T-cell tells the B-cell what to do, the B-cell just goes off and makes its antibody. But of course, all these mutations, VDJ recombination, hypermutation, are creating new receptors for these B cells to capture antigens, and they become numerically and quantitatively the most powerful antigen-presenting cells for CD4 cells in the body. And the trouble is that half of them are plasma cells, CD19 positive plasma cells, because in humans, most plasma cells haven't extinguished the B-cell program completely. They're CD19 positive. The hematologists have known this for 30 years. And so when you come along with anti-CD20, even with an anti-CD20 CAR T-cell, I would expect, you only take out some of these mutated antigen-presenting cells that are capturing autoantigens and creating a feed-forward loop with the helper cells. But if you target CD19, you'll take out almost all of them. And we think that it might be a key to the extraordinary results that George Shett is going to tell us about this afternoon in lupus and other diseases where I think most of us had given up hope that we could ever cure disease. So I better wind up there. And just to say that B-cell tolerance, it turned out to be much more complicated than anyone could have anticipated. And instead of it being a if in doubt, throw it out binary system, it's this very much more nuanced process of waste not, want not, that as David will tell you, don't get rid of a receptor, edit it in the bone marrow. And what I've told you is that this whole phenomenon of energy is really probably all about hedging your bets and finding a way to recycle receptors to undergo clonal redemption and mutate bad antibodies into exquisitely specific good antibodies. But that whole process needs mutations, and playing the mutation comes with risks, and that is probably at the core of autoimmune disease. So I want to come back to Planck's principle and just say that, as I think you've got a sense that for this field, progress really has been not through waiting for funerals, but through births of new technologies. But for the younger people in the audience, what I want to say is that don't wait for my funeral. Grab the latest new technology, because this will change yet again, and show where I had it wrong or wasn't even asking the right question. Because as sure as day, that shoe, Planck's principle to the other foot, but birth of new technologies will straighten it out. Now, very, very many people have contributed to the work that I've skimmed through today. I've highlighted some through there. The unpublished work, you can see the authors there. I thank you again enormously to the Crawford Foundation. To be able to celebrate this process in such a way is just a huge privilege. And thank you to the Royal Swedish Academy of Sciences for choosing this topic, because it is a beautiful topic and a very important topic. And most importantly, thanks to my wife, Suzanne, who has been the most wonderful partner, both in science and in family and in life.